A warm welcome to botany class. Today we are going to move the third chapter, chromosomal basis of inheritance. Third chapter is chromosomal basis of inheritance. In the previous chapter you have learned Mendelian inheritance, Mendelian genetics. Now you are going to study the deviations of concepts related to Mendelian inheritance and chromosomal basis of inheritance. Before entering into the chapter, look into the chapter outline. Chromosomal theory of inheritance, historical development of chromosome theory, salient features of chromosomal theory, comparison between gene and chromosome behavior, linkage, kinds of linkage, that is complete linkage and incomplete linkage. These are the topics you are going to cover today. Let's begin with chromosomal theory of inheritance. You learned genetics very well in the last chapter. Who was the father of genetics? Gregor Johann Mendel. You all know that Gregor Johann Mendel studied well-defined characteristic features of pea plants. He studied very well, but till 1900 it was, or his findings was not recognized or unrecognized. So there are three scientists, Hugo de Vries, Karl Correns and Tesmark, rediscovered Mendel's findings and report it. Various cytologists also studied the cell division due to the investigation of microscope or due to the development of or advancement of microscopy. Various cytologists, they also studied cell division. Due to this, they find the structures which are present inside the cell. You know, in eukaryotic cell, there is a difference between prokaryotic cell and eukaryotic cell. In eukaryotic cell, there is a well-defined nucleus. One of the characteristic feature of eukaryotic cell is well-defined nucleus, which contains nuclear membrane, nucleolus and chromatin reticulum. So due to the discovery of this microscope, they found out there is a worm-like structures found within the nucleus. At the time of cell division, it condensed and formed as a thread-like structures called chromosome. Chromosome. The name of this worm-like structures found in eukaryotic cell is chromosome. These chromosomes carries genes. These genes are located in different position or locus, particular position or locus, and they are arranged linear in the chromosome. The chromosome which contains, here is the chromosome, this chromosome contains a coiled structures called DNA, the structures which contains the genes arranged in linear order. Linearly it arranged. Each gene carries, here there are genes. These genes are arranged in linear order. Each gene located in a particular position. And these genes carry information from one generation to the next generation. So chromosomal theory of inheritance states that the genes are located within the chromosomes. This is the chromosome. Inside the chromosomes, the genes are located. Gene 1, gene 2. So, these genes are located within the chromosome and it carries the information, that is the characters or determiners, which carries these characters from generation to one generation to the next generation. That is the chromosomal theory of inheritance. Then, historical development of chromosomal theory. Various scientists, they discover and postulated different ways. Wilhelm Rox, number 1. Wilhelm Rox postulated that the genes which are transmitting characters, the transferring hereditary character, the characters are carried out or transmit from one generation to the next generation. So, transferring heredity is found out by or postulated by Wilhelm Rox. Then, Montgomery, he suggests that there are distinct pairs of chromosomes. The chromosomes which occurs in pairs. This is one chromosome, this is another chromosome. Now, these chromosomes are named as homologous chromosome. Homologous. Why it is homologous? These chromosomes derived from the parent, one from male, another from female. So, this is paternal, this is from maternal, one from male and another from female. So, these chromosomes are called as homologous chromosomes, the set of chromosomes. So, in eukaryote, the chromosomes are diploid, 2n, diploid in number 2n, that contains 2 haploid chromosomes. So, this diploid chromosomes are called as homologous. How it develops? Each chromosome derived from male and female, from the parent. Now, this set is called homologous chromosome, diploid in number. Then, Boveri. T. Boveri suggests that the chromosomes which carries the determiners, are called as these genes. The genes are known as determiners, different determiners. They determine the character. It was said by Boveri, T. Boveri. And Sutton explains the similarity between Mendelian factors and chromosome behavior. There is a similarity or parallelism between Mendelian factors and chromosome behavior. So, this Sutton and Boveri suggested this or compare or parallelism between Mendelian factors and chromosome behavior. What is the comparison between these Mendelian factors and chromosome behavior? The alleles or factors are occur in pair. According to Mendel said that the alleles, what are alleles? One such example is capital A, or homozygous dominant, or recessive small b. So, this Mendel explained that these alleles or factors occur in pair, but chromosome behavior says that chromosomes occur in pair. That is the comparison. Here the alleles are occur in pairs, here the chromosomes are occur in pairs. Then similar or dissimilar alleles are separate. Either the similar allele, this allele, similar allele or dissimilar, heterozygous, capital A, small b. It separate at the time of gamete formation. Here homologous chromosome separate during meiosis. At the time of meiosis, what happens? At the time of cell division, these pair of chromosomes which contains genes which segregate, which separate. Then these alleles, Mendelian factors, according to Mendel, the alleles are assort independently. At the time of gamete formation, it assort independently, it separate. But here it suggests that the paired chromosomes are separate during meiosis. But linked genes do not assort. What are linked genes? Here in the chromosome, the genes are located very close. Sometimes if the genes are located very close, they inherit together. The genes are named as linked genes. So, linked genes, they do not assort independently, but they tend together, inherit together. This is the comparison between Mendelian factors and chromosome behavior. And in the diploid set of chromosome, all the organisms which possess definite set of chromosomes. What is the, here are some examples. These examples are asked for one word question. So, learn this, there is a tablet column given in the book. The diploid set of diploid chromosome in Ophioglossum. Ophioglossum, one of the pteridophyte, Ophioglossum and Equisetum. So, this Ophioglossum contains the diploid set of chromosome, because each organism which possesses a definite set of chromosome. Diploid set, here what is the diploid set of chromosome in Ophioglossum? 1262. Equisetum 216. Giant sequoia 22. The diploid set 2n. Diploid set of chromosome is 22. Arabidopsis thaliana. In Arabidopsis thaliana 10. Rice 24. Oryza sativa. Potato. Solanum tuberosum 48. Maize. Siam maize 20. Onion. Allium cepa 16. Then Haplopappus 4. So, in this given numbers, the more number is Ophioglossum 1262. Lowest is Haplopappus 4 in number. The diploid set of chromosome is 4 in number. Then let us move to the salient features of chromosome. What are the salient features of chromosome? The somatic cells. The somatic cells are known as body cells. The somatic cells of an organism which is derived from the zygote. At first the somatic cells are derived from the repeated division of zygote. From the zygote divides repeatedly and forms the somatic cells. So, these consist of identical set of chromosome that we called as this homologous chromosome. The identical sets of chromosome is homologous chromosome. One is derived from paternal, another one derived from maternal. Maternal mother, paternal father. So, this complete set is called as homologous chromosome. And the chromosomes, so the salient first feature is homologous, regarding homologous chromosome. First write about the salient features. Salient features of chromosomal basis of inheritance. About homologous chromosome which are derived from maternal and paternal. Then the chromosomes have the structural and uniqueness. Throughout the life cycle of this, there is a uniqueness in the chromosome. There is no change. If any changes in the chromosome, mutation takes place. There is no change. Lifelong it is there. Then each chromosome carries a specific locus or specific position. Each carries a specific locus. This is the locus, or these genes are located in a specific position. And the behavior of the chromosomes. The behavior of the chromosomes during gamete formation. at the time of gamete formation, that is at the time of cell division, meiotic division. These genes are located and these genes, gamete formation takes place at the time of meiosis. So, first tell about homologous pair of chromosome, then the chromosomes retain their structural uniqueness throughout the life cycle, and each chromosome carries a specific position or locus. Salient features, sometimes 3 mark question or 2 mark question, they will ask what are the salient features of chromosomal theory of inheritance. Now, let us move to the next concept, that is linkage. Linkage. Define linkage. Linkage, look at this picture. This is a homologous chromosome. In this homologous chromosome, the genes are located in a specific locus. So, they are called as determiners. What it determine? The characters are determined from this genes, and these are carrying information from one generation to the next generation. Now, linked genes, linkage. When the genes are located together in a chromosome, for example, if you are taking a chromosome, this is a chromosome. This chromosome contains 2 genes. This is gene A, this is gene B. So, these genes or 2 genes are closely inherited, located very close. In this case, it do not assort independently, but it inherit together. The tendency of inheriting to the characters which are inherit together is called as linkage, and the genes are named as linked genes. So, in linkage, normally according to Mendelian inheritance, at the time of gamete formation, these genes, either it is present in the same chromosome or it present in different chromosome which assort independently. It has to separate at the time of gamete formation. But biologists found in certain cases it do not assort independently. So, this that is the reason these 2 very closely arranged genes which are inherited together. The tendency of inheriting together, the genes or the characters which are expressing together are called as linked genes. So, one such examples are observed by Bason and Punnett. They observed in sweet pea. The name of the plant is sweet pea. In sweet pea, the name is the botanical name is Lathyrus odoratus. Lathyrus odoratus. In sweet pea, they observe that the linked genes. So, they are choosing one example. In the character of this is selected as purple flower The color of the flower is purple. In sweet pea, Lathyrus odoratus, purple flower with long pollen. Long pollen grain. Cross with red flower and round pollen. Purple flower with a long pollen, red flower with round pollen. So, the character of this is linked gene. So, in linkage, The characters chosen by Basin and Punnett is purple color flower in sweet pea or Lathyrus odoratus. The color of the flower is purple flower with long pollen and red flower with round pollen. So, the character is the alleles. Capital P, capital L, capital P, capital L. Here, small p, small l, small p, small l. So, these are the parents. The selected parents are purple flower with long pollen, red flower with round pollen. Now, in the chromosomal form, this is the allele. When you draw the chromosome, here is the gene, capital P, here is capital L. Capital P, capital L. Here, small p, small p, small l. Here, small p, small l. This is the allelic form, this is the chromosomal form. So, alleles are located, the genes are located in pairs. Here Mendel represents this as alleles. Now, this character, this is the parent. When you make a cross between these parent, that is purple flower with long pollen and red flower with round pollen, it gives in the F1 generation, it produces all the plants. The F1, normally in the F1 plants are inherit together and it produces a hybrid. So, it produces capital P, capital L. So, this produces, what are the gametes from here? First is taken the gametes out. This is the parent and the gametes. Here capital P, capital L. Here small p, small l. So, in the F1 generation, it produces, this is the gamete. So, it gives capital P, capital L, small l. Capital P, capital L, small l. So, in this, it gives in the chromosomal form, you draw like this. Here capital P, capital L, here small p, small p, small l, small l. So, in the from this gamete, in the F1 generation, it produces this capital P, capital L, small l. This is the hybrid. The character is purple flower with long pollen. It produces in the F1 generation, it produces purple flower with long pollen. So, this purple capital P, capital L, small p, small l, which inherit together and it produces this capital P, capital L, small p, capital L, capital P, capital L, small l. So, it is a hybrid purple, capital P, L, purple with long pollen which dominate. The dominant character is purple flower with long pollen, recessive character is red flower with round pollen. But F1 generation, it produces the dominant character. The expression is dominant character, that is purple flower with long pollen. Now, in the linked genes, according to the normal Mendelian hybrid or Mendelian factors, in the F2 generation, this is the hybrid, F1 generation it produces the hybrid. But F2 generation, when you make a cross between this hybrid with recessive, pure recessive, that is small p, small l, small p, small l. When you make a cross between homozygous recessive or it undergo test cross, the expected ratio is it should give 1:1:1:1:1. The expected ratio according to Mendelian inheritance is 1:1:1:1. But in this case, in this Lathyrus odoratus, it do not give the expected ratio. Instead, it gives a greater number of these parental forms, that is purple flower with long pollen and red flower with round pollen. Either this purple flower with long pollen or red flower with round pollen. So, they concluded that these genes are not assort independently according to Mendel's law of independent assortment. So, these characters or these genes are inherited together. So, there are two different types. One is coupling and repulsion. Coupling and repulsion. In coupling, if the dominant genes and the recessive genes, dominant gene, here there is a chromosome, in this chromosome, a homologous chromosome, here the dominant gene, either capital A and capital B or small a, small b. The dominant coupling, the genes, if the dominant and recessive genes arranged in the same homologous chromosomes which inherited together or tend to be inherited together and gives the ratio 7:1:1:7. So, here the recombinant frequency is very less. These two are recombinant genes, but it increases the parental form. This purple flower with long pollen or red flower with round pollen. That process is called as coupling or cis configuration. Cis configuration. The very important 2 mark question. What is meant by coupling and or what is cis configuration? What is meant by coupling? If dominant and recessive alleles are present in the same homologous chromosomes which tend to be inherited together and it increase the parental forms, it gives the ratio 7:1:1:7. So, these genes are called as linked genes. And repulsion, what is meant by repulsion? When the dominant and recessive genes are located in the different chromosome but in homologous chromosome. Look at this diagram. Here is a homologous chromosome. This is homologous chromosome which contains capital A, small b or small a, capital B. So, this is one homologous chromosome, this is another homologous chromosome. Here the dominant and recessive alleles are present in the same homologous chromosome. Here dominant capital A, capital B present in same chromosome, small a, small b present in same chromosome. But here dominant and heterozygous forms. So, when the dominant and recessive alleles present in different chromosomes of different homologous pair, it inherit far apart. That is repulsion. Coupling, it inherited together. Repulsion, it far apart. It inherit far apart. And it gives the ratio 1:7:7:1. So parental forms, it greater amount of parental forms. Recombinant forms are less. So this is called as repulsion, coupling or cis configuration, repulsion or trans configuration. So this question is very important. Linkage. What is meant by linkage? Coupling and repulsion. Let's see this coupling and repulsion in the same example. See here, the color of the character is color of the flower and size of the pollen grain, shape of the pollen grain. In this coupling, first coupling in the F1, when make a cross between these two, purple flower with long pollen, red flower with round pollen. In the F1 generation, these are the parent, and take out the gametes, capital P, capital L, here small p, small l. So in the F1 generation, it produces capital P, capital L, small l. That is purple flower with long pollen, because F1 generation, it produces a hybrid. This is a hybrid. Now this hybrid undergo test cross. What is meant by test cross? A hybrid with recessive, pure recessive or homozygous recessive, small p, small l. When you make a cross between this, what is the expected ratio? The expected ratio is 1:1:1:1. But in this case, Lathyrus odoratus, when Bateson and Punnett experimented this, they do not expect, or it do not give the expected ratio 1:1:1, but it gives the ratio 7:1:1:7. That is the coupling. How it gives? This produces, here there are, take out the gametes from this in the F1 generation. What are the gametes? Possible gametes, capital P, capital L, capital P, small l, small p, capital L, small p, small l. So draw it in the form of chromosome. Here two, 3, 4. So here it gives capital P, capital L. So here capital P, capital L. The next one, capital P, small l. Capital P, small l. Then small p, capital L. Small p, capital L, and small p, small l. Small l. Possible gametes are 4 possible gametes. Cross with this small p, small l. Small p and small l. Now it gives the combination of this and this, this and this, this and this, this and this. So it gives the ratio 7:1. Expected ratio is this one, 1:1:1:1. Instead of this ratio, it produces 7:1:1:7. This is coupling. So it possesses capital P, capital L, small p, small l. 1 here and next here. and here it gives capital P, capital L, small p, small l, small l. Then here it gives capital P, small l, small l, small p, small l, and here small p capital L, small p capital L, and small p small l. Then last one, it gives small p small l, small p small l. So the parent form purple flower with long pollen, this one P, capital P, capital L. So this and this parental forms, it increases, and these two are the recombinant forms. So linked genes, here this coupling ratio is 7:1:1:7. Then repulsion. When you take the example for repulsion, here the parent is purple flower with round pollen. Here in repulsion, you take the parental character as purple flower with round pollen. Make a cross between red flower with red flower long pollen. So here the character is purple, capital P, purple flower, capital P, and round pollen, small l, capital P, small l, into red flower with long pollen, small p, capital L, small p, capital L. This is the character or parental form which is taken in repulsion. So in repulsion, this purple, this is the parent. These parents take out the gametes from here. When you are taking out the gametes, it produces capital P small l. This is one gamete into small p capital L gamete. So in the F1 generation, it produces capital P small p capital L small l. This is the hybrid in F1 generation. In the F1 generation, it produces a hybrid. When you make a cross between test cross, when it undergo test cross, that is pure recessive, small p small l, it gives the ratio. When you make a cross between this, what are the combinations? Here gametes, capital P capital L, capital P small l, small p capital L, small p small l. 4 different combinations are there. Capital P, capital L, capital P small l, small p capital L, small p small l, and cross between small p small l. So when you make a cross between these two, it gives the ratio. When you make it like this, it produces the ratio 1:7:7:1. So this ratio is called as repulsion or trans configuration. Coupling is cis configuration. This is trans configuration. I hope that you understand this concept very clear. Once again, you recall what you learned today: chromosomal theory of inheritance, historical development, salient features, comparison between gene and chromosome, then linkage. Then there are two different types of linkage: complete linkage and incomplete linkage. The kinds of linkage is two, which is classified by Morgan. Morgan classified two different types of linkage: complete linkage, incomplete linkage. What's the meaning of complete linkage? Complete linkage, the genes are located very close. The genes in the chromosome which located very close, so which is determined by the frequency of recombination. Recombination frequency is, if the recombination frequency is more, it gives this recombination frequency, if it is more, it produces unlinked genes. If the frequency is less, recombination frequency is less, which increases the linked genes. So complete linked genes, the crossing over is do not exhibit, because the genes are located very close, so it forms complete linkage. The genes tend to inherit together. So there is no exhibition of the crossing over, that is new combination. But incomplete, it exhibit crossing over, because these genes are located far apart in the chromosome. So there are two different types of linkage: complete and incomplete. Draw all these diagrams in the class note and learn all these questions. Thank you.